This is Midwest Moxie, a show about our region's visionary entrepreneurs and the experiences that shape them. I'm your host, Kathleen Gallagher, Executive Director of Five Lakes Institute. Mike Schultz had spent more than a decade in his lab at University of Iowa discovering a pair of radioactive substances that could detect and destroy cancer cells. Fran Johnson, his wife, had spent just as much time running the University Medical Center's heart disease treatment program. So when they launched a startup in 2015, the division of work was obvious. Mike stayed at the university to continue his research, and Fran left to run the company. Viewpoint Molecular Targeting raised more than $50 million of grant dollars and investor funding, and in 2023 merged with Isoray to form a new public company called Perspective Therapeutics. It's headquartered in Seattle and has about 1/3 of its employees in Iowa. Mike and Fran, welcome to Midwest Moxie.
Thanks, Kathleen. We're just delighted to be talking to you today.
Likewise. Radioactive substances had been used for some time as contrast agents for medical imaging. But in the early 2000s, you started trying to find molecules that would not only help the imaging, but also destroy cancer cells and other diseased tissues. What inspired you to do this pioneering work?
The inspiration really came from a telephone call that I had from a physician scientist who was the head of nuclear medicine at the University of Maryland in Baltimore, and his name was Bruce Line. He was a physician really interested in the idea of taking this concept of targeting radiation to tumors and transforming it, not only using it as a diagnostic tool, but also precisely delivering radiation to tumors for therapeutics. And I had been working on some radioactive isotopes in my research at the National Institute of Standards and Technology in Gaithersburg, Maryland. I went to his laboratory, and the first experiments that we did clearly showed that the use of alpha particle emitters for cancer therapy could be advantageous compared to beta emitters. And that just led to a whole career of working on ways to deliver radiation to the tumor microenvironment.
So really, you already knew that you could use these radioactive substances to see the cancer. And what you wanted to do is find a pair for the part that could see that would destroy the cancer.
Absolutely. And sort of the natural history of that is we started looking at lots of different ways to be able to deliver the radiation to the tumor microenvironment. We started with what are called monoclonal antibodies, for example. These are sort of natural biologics that you can develop to bind very specifically to proteins on cancer cells that are not on normal cells. But as we learned more about that, it turned out that they had circulation times in the body that were too long for delivering therapeutic radioisotopes.
You don't want radiation to stay in the body for too long.
You absolutely don't.
Yeah.
And so eventually we came upon these, what I call peptides and peptide-like small molecules that have very fast pharmacokinetic properties. That means that they deliver the radiation to the tumor microenvironment very quickly, and then the residual compound, stably coupled to the radioactive isotope, is cleared from the body very quickly through the renal system into the bladder. And so that reduces any of the potential for off-target effects that could happen and produces a better tumor to normal ratio that gives you better therapeutic benefit.
So you weren't just looking to destroy the cancer. You had to develop this whole system for how to get it there, how to get it out quickly. I love the name of this. This area of medicine is known as theranostics, a combination of the words therapeutics and diagnostics. You got to love that. So you found two forms of lead that would help image and destroy these cancer cells. Isn't lead toxic to humans? Why lead?
So that's a great question, and there's lots to unpack there. But the way it really worked was we were trying to optimize the isotope for the pharmacokinetic properties that we found for the delivery vehicle. And it turns out that lead-212, which delivers alpha particles, has a half-life that matches up better with these molecules that go to the tumor really quickly and are washed out of the body very fast. And as it turns out, lead-212, the therapeutic isotope, there's an isotope of lead, lead-203, that can be used for the diagnostic part. So that way you can deliver a gamma-emitting isotope for imaging the disease, and then you can use that to develop the therapeutic strategy for delivering the therapeutic And because lead-212 and lead-203 are elementally identical, you know that their biochemistries will be identical. And so that way you can depend on that imaging agent and to predict the therapeutic. And this is the only practical isotopic pair that can be used for this. Now, in terms of your question about toxicity, these are radioactive lead. So first of all, they decay to something else. But the reality is that this is a tiny amount of lead. It's below the limit of detection. It's about the same amount of lead that you might find if you added up all the lead that was in 500 bottles of drinking water.
Wow. So, Fran, he must have convinced you that putting lead into people's bodies was a reasonable thing to do. You had been running this large clinical program at University of Iowa. What made you decide to go and run that startup?
Well, Kathleen, that's, I guess, a pretty valid question. But I was section head for an organ failure program at a tertiary medical center, and I had functioned in that kind of a role at 3 different medical centers over a period of approximately 20 years. We had, 1,000 to 1,200 patients under management at any one time, of whom about 25% would die in a given year.
Oh.
So my job was really about providing these people for whom standard of care had been ineffective with access to new drugs and devices that would prolong their lives. And so my decision to put my efforts behind this radiopharmaceutical development was just an extension of my personal mission to bring cutting edge therapies to the patients who need them, and spoke to the preclinical strength of the evidence that I was seeing from Dr. Schultz's lab. I was completely convinced that this was going to be a revolutionary new treatment platform. And I was also convinced that it would require both public and private investment in order to move it forward in a meaningful way. I wanted to see a drug approval, and you can't do that staying in academics.
I just want to say, when you said Dr. Schultz's lab, you were talking about your husband in a professional manner.
Correct. We always refer to each other as Dr. Schultz and Dr. Johnson in the workplace.
So, you were running this big clinical program, and some might say, well, how would she know what she was getting into with a startup? But you actually had been a co-founder of another biotech startup when you were an assistant professor at Stanford. How did that experience help prepare you to run Viewpoint Molecular Targeting?
Well, my first company co-founding effort was in a personalized molecular diagnostic space for transplant recipients and predicting transplant rejection. And so it was a very different time, different technology platform, different geography than our radiopharmaceutical company. But nonetheless, I did learn some very important lessons from that first startup. And I think the most important concept was the time-money equation constant. Money times time times a constant equals productivity. And the constant is different in business than in academics. You must move much faster. You have to have productivity that leads to changes in patient outcomes. And so, that really dictates what you can accomplish, with private money in a particular amount of time. And so I was far more realistic about what sort of approach we needed to take in order to develop this. We were very much bootstrapped. And early on, what we did is we relied heavily on government grants, the Small Business Innovation Research Grant Program, which Mike can tell you more about.
Or SBIR is what a lot of people call them.
Yes, SBIRs. And that moved, I would say, at the pace that our science could move forward, because we wanted to accomplish a lot. It was going to take us a while. And we paced out the ratio of public to private funding to meet our goals. And when we got to a particular change in what we thought our valuation as a company would be, we would go out and we'd try to raise more. And I don't think that I would have had the ability to gauge and change our strategy the way I did if it hadn't been for that experience.
You are in a really hot area now. When you started, there were maybe a handful of radiotherapeutic companies, and now there are more than 100. You've got 50 plus employees in Iowa in an R&D center and a production facility in Coralville, outside of Iowa City. Tell us about where you're going from here. How big can this company become, and what other facilities do you need to do it?
It made a tremendous amount of sense for us to keep the Discovery Center and our early production facilities near Iowa City, near the University of Iowa, where we spun out. I had a group of former students who now have 10 years of experience or more in this area who are now leadership on our discovery team. And as Fran mentioned, we were a bootstrap company, so very, very conscious of our burn rate. And at the earliest stages of development, keeping discovery in Iowa was guided by that sword, right? So financially, it didn't make sense for us to move at that time. And the more we grow, the more we get entrenched there. And it turns out that the biosciences program at the University of Iowa, there's $800 million or odd dollars of NIH money that goes through the biosciences program at the University of Iowa every year. So biomedical engineering and electrical engineering and chemical engineering, all these things factor into the development of our company. And the University of Iowa is a terrific place for us to be able to recruit from. And people who live in Iowa City and in that area of Iowa tend to stay there. It's a great place to be. So that just added to our intentional keeping the company based in Iowa. That said, we, as I mentioned earlier in our conversation, lead-212 has a relatively short half-life. And so that means that we'll have local, regional, what I call manufacturing and distribution of the final drug product. So we have publicly disclosed that we have facilities that we're building out in Chicago and Los Angeles and Texas to meet the needs of cancer patients across the United States. And so there will be build out in those areas to accommodate manufacturing and distribution. But we see, clearly in the future that the anchor point for the company will be in Iowa.
You pioneered this area of radiotherapeutics. What does it feel like 20, 25 years later to see how it has grown and what you started out doing?
Yeah, it's pretty remarkable. I think there was one or two therapeutic radiopharmaceuticals that, formed the basis for my research in this area in early 2000s, let's call it, when I met with Professor Line at University of Maryland in Baltimore. And it's remarkable to see the kind of progress that we've made. And it's been an inspiring thing to me to see the number of companies that now have grown out of these ideas that I think that we really did pioneer all those years ago, especially in the area of alpha particle radionuclide therapy for cancer. And this really, truly does have the potential to be transformative. And the kind of interest that has sprung up around the world in this area is truly remarkable. And we're kind of proud to have been people that believed in this from an early time, and seeing it and the kind of impact that it's had on cancer patients is really is rewarding in and of itself.
And it started right there in Iowa. Talk about Midwest Moxie. Thank you so much for joining us, Mike and Fran.
You bet. Thank you.
You can read more about today's guest by visiting wuwm.com. Don't forget, you can also listen to the show as a podcast if you'd like to take us on the go. Just search for Midwest Moxie, that's MOXIE, wherever you listen to your podcasts. I'm your host, Kathleen Gallagher. Midwest Moxie was created by executive producer Audrey Nowakowski and me. Today's episode was produced by Audrey Nowakowski and engineered by WUWM's Jason Reevy. Join us next Saturday afternoon at 2:30 to hear from more visionary Midwestern founders building innovative companies. That's right here on 89.7 WUWM, Milwaukee's NPR. Support for Midwest Moxie comes from Microsoft, whose efforts to drive technology development across the Midwest and beyond include its National TechSpark community engagement work, which started in Wisconsin, its role as co-founder of Titletown Tech in Green Bay, and its new AI co-innovation lab at University of Wisconsin-Milwaukee. More information is available at Microsoft.com. Support also comes from Quarles, a multidisciplinary AM Law 200 firm with approximately 550 attorneys who support large and small businesses, from Fortune 500 companies to startups and entrepreneurs across the country. More at Quarles.com.